everyone. Thanks for the great opportunities and invitations to introduce my research. So I'm Joyce Chen. I'm currently an assistant professor at Pritzker School of Molecular Engineering and joined the positions with BME Department for Cancer Research and University of Chicago. And I'm very excited to talk about our researches in my current lab and have some extension work from my postdoc periods with Dr. Varmus. So the main biological technologies in my lab is focused on the stem cells. We study the stem cell biologies, mechanisms, as well as we developed the technologies for disease modelings and for dating of the stem cells biology. So the recently advanced in stem cell technologies, especially the generation of the human lineage cells, through directed differentiation of human pluripotent stem cells, including human embryonic stem cells and induced pluripotent stem cell. They provide us a rigorous human systems unable to modeling and studying human cell biology and disease using the human origin resource. So the main focus in my lab is on the lineage cells. Basically, on the previously published protocols derived in Hans Schnicker's lab at Columbia Universities, we were able to extend these protocols and modify the establish the lineage development and differentiations in my lab. So we firstly differentiated the human pluripotent stem cells into the definitive endoderm and narrow to the anterior foci endoderm. And these endoderm cells further differentiated into line progenitor cells that are triple positive for lineage marker NKS 2.1 and the endoderm stem cell markers FOXA1 and the early line progenitor markers SOX2. And our protocol are capable to generate the line progenitor cells with the purity about the 90% on the culture dish. These line progenitor cells are able to form these 3D organotypic structures on top of the matrix. And our protocol further capable these cells differentiate into either proximal or distal line lineages, including alveoli type 1, type 2 cells, club cells, basal stem cells, ciliated cells, all those major cell types. Our protocol is also capable to generate some real cell populations in airway epitheliums, such as pulmonary endocrine cells, tuft cells. So the further question is how similar our cells compare to their native counterparts, right? So we use the traditional methods such as immunostaining, FACS, as well as single-cell RNA transcriptomics analysis, further indicate the great similarity between these human pluripotent stem cell-derived lung cells with the native ones found in human body. And this single-cell RNA-seq data we use to compare with our data is derived by Mark Krasnow's lab at Stanford University. So as you can appreciate this highly correlation between our cells with these real native ones in human lung system. So these are all great cell resources derived from pluripotent stem cells and from a human origin. And these are great resources provide us for modeling and studying human lung disease in these platforms, and especially use this real the human cells system. So next, we're going to demonstrate one of the main applications in my lab to use these pluripotent stem cell-derived lung cells for studying small cell lung cancer. So why is small cell lung cancer? So why the question should be why does small cell lung cancer need novel approaches or new model systems? Just give a brief background about small cell lung cancer. Small cell lung cancer is the most aggressive type of the lung cancer with a 5-year survival rate less than 5% and leads to the loss of the 30,000 lives per year in the United States. There is a very interesting characteristics about small cell lung cancer. Okay, especially the small cell lung cancer genome has a very high mutation rate, with average 5.5 mutations per megabase pair, and the inactivation in both tumor suppressor genes, RB and p53, are nearly universal, occurred in every case. And there's no big improvement in diagnosis or therapies during the past 3 or 4 decades, indicating our limitations in understanding precisely the disease mechanisms. Especially, it is very difficult to study the small cell lung cancer initiation and progression, mainly due to the scarce of the clinical specimen at the disease early on intermediate stages, because most patients at the diagnosis already developed the metastasis disease, and the surgical removal is really not the right options for those patients. So this results in a shortage of the clinical specimens at the early or medium stage disease, and for the mechanistic studies, for the cancer cell line development, and for those genetic therapeutic screening. Since it's difficult to obtain the clinical specimens for small cell lung cancers, and for develop the cell lines or xenograft for studies, so we turn our focus to make this model system for small cell lung cancer. So first, we think about whether we can transform these normal cells into cancer cells at the various stages. So the pulmonary neuroendocrine cells is believed to be the cell origin for small cell lung cancer, and this hypothesis has been initially proved by Antenberg's group with his research published at Cancer Cell. At that research, they first introduced the both RB and the p53's loss function mutation under specific cell lineage promoters using these Cree and LOPS systems. So they use this Cree DNA recombinants under at least 3 cell types: alveoli type 2 cells, pinna cells, as well as club cells. And their work indicate only pinna cells served as the best cell origins reach out to the best effective transformation rates for these cells to become the early stage of cancerous cells with the resembling morphology to small cell lung cancer in mouse model. So the pinna cells are believed to be the positive cell targets for small cell lung cancer initiation. So we give a brief introduction about the pinna cells. So pinna cells existing in airway epitheliums as a cluster called the neural epithelial body, as well as single cells. And these cells are uniquely express the biomarkers such as secretory proteins or peptides, including CGRP, SIMP, PGB 9.5, has specific markers NCAM1 and transcription factor SCL1. So pinna cells is believed to play multiple roles in the lung function, and especially pennant cells is believed to be a progenitor-like cells contributing to the lung development in the branch morphogenesis, regulate the lung cells growth and maturism. Especially these cells also contribute to the lung repairs after the entry. So then so now we look at whether we can, extract or culture out the native pennant cells for, small cell lung cancer disease modeling. However, it is extremely difficult to extract the pennant cell out because pennant cells is a very real population, counting for only 0.5% of the total airway epitheliums, and there's no robust systems or culture protocols for further maintaining and expanding the pennant cells in vitro in culture dishes for the in-depth analysis. So then we turn out our attentions to these pluripotent stem cell-derived model systems. In the previous protocols provided by others in the field, to generate a abundant number of the pennant cells in the culture system. So then we decided to modify these protocols mainly by blocking the notch signalings. We suppressing the notch signaling, particularly during the lung differentiation phase from the lung progenitor cells to the differentiated cells. And these modified protocols successfully provide abundant pennant cells in our petri dishes. These cells accounting for around 8 to 10% of the total cell population, and the cells actually express all the main markers reported in the native pennant cells, including secretory proteins, CGRP, PGP9.5, NK1, as well as the lung lineage marker NKS2.1. Further characterizations indicate our cells actually express all other markers reported in the native pennant cells, including chromogranulases, NK1s, and transcription factor SCL1. And the single-cell RNA-seq profiles further validate the highly resembling of our cells to the native counterparts, as these pluripotent sensor-derived pennant cells expressing nearly all the biomarkers reported in the native ones, including some secretory proteins, peptides, neuron transmitters, as well as some regulatory factors to regulate the epithelial cells or immune systems, indicating these cells, even derived from pluripotent stem cell, are fully physiologically functional. Okay, so now we have these pennant cells, fake pennant cells, but it's very likely the native one. So we have this great resource, and we think whether we can use these cells for modeling small cell lung cancer. So the first cancer genes we think of to manipulate is RB genes, because RB loss is highly associated with neuronal type of the tumors, and RB loss actually is a universal event occurred in nearly every small cell lung cancer patient case. So we use this doxycycline inducible system to particularly make the RB loss in the lung differentiation phase. And our methods by RB loss dramatically increase the pennant cells number in the petri dish. As you can see, the pennant cells proliferation rate of cell numbers increased from 10% to 40%. And this is resembling an early stage of the oncogenic transformation phenotype, as these cells actually undergoing oncogenic transforms or hypoplasias in the petri dish. However, the main hallmark for cancer development is to form these real tumors in vivo, right? So we testing whether our pennant cells are capable to form the tumors in immune deficient mice. So we first introduced the peanut cells with only RB lost. However, nothing happened. So these cells failed to develop the xenograft in immunodeficient mice. Then we started to introduce the second mutation in p53. So our cells with both reduction in RB and p53 successfully developed this tumor-like xenograft growth in mice. Through further characterizations, we found these xenograft tumors is highly resembling the early stage of the tumors found in clinical small cell lung cancer case. And through the further molecular cellular characterizations, we found most cells express the lung lineage marker NKS 2.1, as well as the typical peanut cells or small cell lung cancer markers, such as they express dysfunctional peptides, CGRP, and all other, clinical markers like NCAM1, CD56, as well as transcription factor SCL1. So then our results indicate these pluripotent cell-derived peanut cells are capable to form the tumors that are highly resembling small cell lung cancers, use these commonly mutant genes found in small cell lung cancer. So indicating this model system is capable to recapitulate these main features in small cell lung cancer at the early stage. So then we published our initial data in JEM paper in 2019. After our paper published, we received lots of the questions from this field, especially from the small cell lung cancer field. And one of the main questions is, oh, that's great, you can show this pluripotent cell-derived peanut cells are capable to form the early stage of small cell lung cancer. But what about the middle stage or late stage, right? Are your cells fully capable of mimicking the various stage of the small cell lung cancers, especially this highly malignant property of this type of the cancers? So then we decided to extend our protocols to introduce the third mutations called MYC. So MYC mutation actually accounting for around 20% of the total small cell lung cancer cases. And under MYC overexpression, these cells actually turn to the malignant transformations and eventually develop this highly advanced phenotype and metastasis. So then we decided to introduce three mutations in our cell systems, p53, RB loss, and overexpression of MYC. So first, we still using the inducible system to put these three gene mutations in the system and only turn on these three mutations specifically at the lung differentiation stage. And then we inoculated this mixture of the lung cells in the immunodeficient mice. And the second strategy is we specifically purified these peanut cells out using a reporter systems. We put these YFPs under a peanut cell specific lineage marker CGRP promoter, and to mark these cells with YFP. And these strategies will allow us to isolate these peanut cells in life and for follow-up study. So both strategies either inject the mixture of the island cells, or either we purified these peanut cells along with three gene mutations. Both approaches capable to form the small cell lung cancers at the middle or later stages. And these cells, especially when you inoculate the cells under skins or under the renal capsule, these cells can develop more advanced phenotypes under the skin, and especially when you have this primary tumor formation under the renal capsule, these cells are capable to migrate outside the kidney and develop the metastasis at the distant locations, including liver, intestine, as well as these cells are able to migration or homing back to the lung locations. At this stage, we are very excited about, these painted cell-derived from pluripotent stem cells are fully capable to mimic these various stages of the small cell lung cancer development. And these model systems derived from a genetically defined human cell background, and we can isolate these tumors either from primary location or from metastasis location. This will provide a lot of the information about how these cells can progress from early stage to later stage, and eventually how these cells and develop their metastasis capacities. And this is a great platform. We're able to do a lot of work on that, not just for mechanistic studies, as well as for genetic or therapeutic studies. So first, we characterize these cells derived from primary locations as well as the metastasis locations, including liver and the lung. And we use this single cell RNA-seq data to cluster our cells. We found the cells derived from primary and metastasis location can be divided into at least 3 clusters. We indicate the cells from here is cluster 0, 1, and 2. So first, we wanna take a look at what kind of specific marker expressed by these cells, and our results demonstrate all these cells are express typical small cell lung cancer markers, including their variants. For example, some portion of the cells express the classical early stage marker is SCL1, and they actually progress into the variants indicating the advanced stages. As those cells started to present their variance markers, neuron D1s, YAP, PAL2F3, and they also typically express small cell lung cancer clinical markers MCAN1, and of course, majority cell express MYC because they are overexpressed makers by artificially. I want to point out that there's one similarities between our model system with these mouse models reported recently by Trudy Oliver's lab. They indicate by overexpress of MYC, they can drive this temporal evolution of the small cell lung cancer from early stage SCL1 positive cells into the neuron D1 or YAP positive. And our model systems derived from human cells mimic these evolutions presented in mouse. And first, we try to explain why these cells can present this highly malignant phenotype. So recently, publication by Charles Luden group at Memorial Sloan Cancer Center indicated the small cell lung cancer cells express PLCG2 marker intended to present a high malignancy phenotype. So we look at whether our systems express PLCG2, and we found a lot of the majority cells actually in our system either in primary location or metastasis express PLCG2. So this is great. However, this is still not sufficient to explain why these cells develop the metastasis abilities. So then we started to look into it, and we found a very interesting phenotype is among these 3 major clusters. Here, cluster 2, they count around 1% of the total cell populations in the primary locations, and eventually largely amplified into around 30% of this metastasis location. This actually indicating this cluster 2 served kind of like the driver cells for initiate the small cell lung cancer metastasis. Then we reach out to test the hypothesis whether these cluster 2 populations served as a cellular driver to make this small cell lung cancer progress into more malignant phenotype. So first, we performed this cell trajectory analysis. Our data indicate the cluster 2 served as a stem of progenitor-like cells that give rise to either cluster 0 or cluster 1. So indicating these cells might serve as a cellular origin for these tumor formations in both locations. The second is we wanted to test whether this cluster 2 has a specific genetic or molecular programs that associated with cell metastasis behave. So first, we want to look at this cancer stem cell-like signaling pathway programs within these cluster 2 populations. Interesting, compared to the other 2 clusters, either in the primary location or metastasis location, cluster 2 cells populations are highly enriched in stem cell-related gene or signaling pathways. As we demonstrated here, is cluster 2 are highly upregulated in the Wnt, Notch, MAP kinase, PI3K kinase pathways, as well as those VEGF-up pathways. And this indicates cluster 2 is highly active in this kind of like cancer stem cell like populations or transformation. Yes, so next we'll try to look at whether these cluster 2 populations is highly correlated with other important metastasis-related activities, including EMT, epithelial mesenchymal transitions, invasion, cell migration, and chemotaxis. So interestingly, our gene correlation or analysis results indicate the cluster 2 cells are highly enriched in the metastasis-related programs. Like these cells are activate in highly expressed in the epithelial mesenchymal associated genes, and these cells are capable to produce these extracellular remodeling enzymes and actively involved in extracellular remodeling degradation, as well as the cells are able to migration outside of the primary locations to the distant organs, and the cells also present activation in chemotaxis. So this is all indicate cluster 2 cells served as a activated populations to leading to the metastasis of the small cell lung cancer. So then we started to look back our single cell RNA-seq data and ATAS data. We narrowed to some molecular drivers that are highly associated with these cluster 2 populations. And we found that these cells are uniquely express the transcription factors CHD families. CHD family is our chromosome remodeler family. They serve as a transcription factors to turn on a network associated with a metastasis or stem cell like programs. And also these cells are correlated with a highly expression in CD91 marker. So all these cells are double positive for transcription factors CHD2 and CD91 are served as a molecular drivers. If you compare the cells at the metastasis stages with primary locations, these moleculars are highly expressed in the cells at the metastasis stage. So then the next question we try to answer is whether we can find this the similar counterpart or the similar population of the cluster 2s in the real clinical small cell lung cancer patient sample. So we look back to the published data by other groups, including single cell RNA data by Charles Luden's group, as well as other data using bulk RNA-seqs or the TRIP-C. We found these CHD2 or CD91s all highly correlated with cell metastasis behave, as we see, these markers are highly expressed in the metastasis stage, but not in the primary tumor. We also further correlated with this CHD2 expression with the patient survival data, indicating CHD2 high cells, CHD2 high tumor case intended to have a low survival rates in small cell lung cancer patient. Okay, so I'm going to wrap up here, and here is a very important acknowledgement section. So I actually established my lab at the end of the 2020 at University of Chicago, and for partial of the important work was done in my postdoc lab at Varmus lab at Weill Cornell, and I particularly think for Dr. Varmus' mentoring and resource for developing these model systems. And I was also lucky to recruit a very motivated and productive researchers, including my 3 postdocs, and 1 graduate student and the 2 technicians. And I also particularly think our collaborators, without their support and guidance, none piece of the work here can be done. And finally, I want to particularly thank our funding resource. Yeah, thank you, audience. Yeah, thanks.